Hi, and welcome to part 3 of Heredity and Variation. Today, we're going to be doing past paper questions on this topic. Just a quick note, only proceed with this video if you have already completed parts 1 and 2 and have all your notes prepared and you came fully prepared. All right, that's the best way to make use of this particular video. All right, so I hope you guys follow the instructions, and let's get started. Let's start off with a few multiple choice questions that normally come in the exam, the very popular exams. When you're doing past papers, sometimes you bounce up a question that we may not have done the theory for. That's okay. That will happen in any subject, right? It's impossible to cover 100% of a particular topic. There's always these little gaps and whatnot, and that's where the past papers come in. So let's start with the first one. Among the following pairs of conditions, which pair are sex-linked? Well, when they say it's sex-linked, it means it has to do something with the X or Y chromosomes. Right, remember the last pair of chromosomes, the XY chromosomes, determines the sex of the individual, whether they are male or female. Females have two X chromosomes, but men have an X and a Y. Okay, so which one of these? Well, they're talking about genes, right? In the X and Y chromosomes, remember there will be genes within the X and Y chromosomes, right? There will be genes inside of them, okay? Some of these genes within the X and Y chromosomes cause certain conditions. So which pair is caused by genes on the X or Y chromosomes? Well, this one I don't expect you to know. It's one of those questions. It's either you know it or you don't know it. And I know we didn't actually cover this in parts 1 and 3. So the answer for this one is C. And this here is an explanation here, right? Hemophilia and red-green color blindness are both examples of sex-linked traits. These conditions are typically associated with genes located on the X chromosomes, right? And in case you can't remember what hemophilia is, it's basically when you bleed. Usually when you are bleeding, the bleeding stops because the blood clots. Some people have blood that does not cut properly. So whenever they get a cut, it bleeds for a very long time. So the answer for this one is C. Let's move on to the next one. Another multiple choice question. Which of the following describes the genotype for a characteristic written as capital A, common A? Remember, capital A will be the dominant version of the gene, and the common A would be the recessive part of the gene. So here there are two types, two different versions of the same gene, capital A and common A. We all know that this one is what? Heterozygous. So the answer for this one is B, right? Heterozygous. If I'm just saying, if it was capital A and another capital A and both of them were capital, then the answer would be homozygous dominant. And if it was common A and common A, because both of them are the same again, the answer in a case like that would be homozygous recessive. But the correct answer for this particular question is B. So let's move on to the next one. Oh, I highlighted the answer there. Which of the following sentences accurately describes an allele? Well, let's see. Read it through. Obviously, you can pause the video and try it. But the answer for this one, I'll give you a little definition here of alleles here. The answer would definitely be B, because I just kind of spoke about it. It's alternate forms of a single gene, right? A pair of genes that are basically competing to do relatively the same function. Which of the following is an example of discontinuous variation? Discontinuous is when the outcome doesn't have definite categories. So let me give you an example. So for things like, well, I'm talking about continuous. Let's forget it this here. Continuous, which is the opposite of discontinuous, is on a spectrum. So things like height. Height is a spectrum. There's no definite height for someone. Arm length, the length of someone's arm. Skin color. Those things are continuous, but they are asking for discontinuous. So discontinuous is when you have definite categories. It's either it's this, or possibly something else. It have no in-betweens. And of course, that's gender, right? When it comes to gender, it's either a male or female, right? There's basically two categories, right? And some other examples of discontinuous is eye color. It's either you have black eyes, brown eyes, right? It comes into, well, green eyes. It comes into distinct categories, right? Handedness is either that refers to left hand or right hand, right? Right? Well, I don't know if you all know what a lobe. This is part of your ears. Some people have lobes on their ears, and some people do not. Distinct categories. Those are discontinuous. Right. So oh, this is a structured question, right? So I can't remember which year this is, but let's go through this one here. Sickle cell anemia is caused by a change in the gene which controls hemoglobin. All right. According to this question, they name the gene with two letters. So capital H, capital N is one gene, right? This is one. They didn't use one letter. I don't know why they're using two letters, right? So this one, how it has all capital letters, this obviously has to be the dominant gene. And it is HN is the unaffected allele of the hemoglobin gene. And HS, common H, common S, is the changed gene of the hemoglobin gene. So the common H and the common S is obviously the recessive, right? Remember, the recessive is the weaker one and the dominant is the stronger one. If the genotype is capital, is a mixture of the capital and the commons. In other words, it's a mixture of the dominant and the recessive. In a case like this, we know it's hetero, right? But this is a heterozygous situation where it have two different types. One is dominant and one is recessive. But you call this sickle cell trait, meaning that this person has normal blood by the way, right? Remember, it's the common HS1 that's pushing for the sickle cell in this case. But if a person has the dominant and the recessive at the same time, the body is going to choose the dominant one. And the dominant one here is the good one, meaning that this person will have normal blood and not express any signs of sickle cell in their body. However, the person is still carrying the gene that is responsible for sickle cell disease. It's just that the body chooses not to express that. You call that a sickle cell trait, meaning that this person has normal blood, but he's carrying around the gene that could cause sickle cell disease. And believe it or not, if this person were to have a child with someone else, and that someone else also had an HS, and both HSs were to be introduced into the offspring, then that child would have sickle cell disease, right? So a sickle cell trait is when you are just carrying around the gene, but not expressing the symptoms of that particular gene, not symptoms, the trait of that gene. But however, right, what is one advantage of sickle cell trait? You might think that's a kind of bad-ish thing, not a totally bad thing, but a bad-ish thing. But there's at least an advantage of being a person who has sickle cell trait, meaning one dominant and one recessive. It actually it gives some resistance to malaria. Believe it or not, the disease that's associated with mosquitoes, right? It gives some resistance to malaria. Well, I already put the answer for part 3 here. Let's go through it though. Yeah, it wasn't supposed to be on the screen, but it's on the screen. The answer for part 3 is there. So let's go through this, right? According to the question, right? There was a man with capital HN and common HS. Look at here, capital HN, common HS. He's heterozygous. And there's a woman with normal hemoglobin. When I say the person has normal hemoglobin, it means there's no sign of the sickle trait. So what they're really trying to say is the woman has two good dominant genes. She is homozygous dominant. And these here are the outcomes, right? In this square here, you have two capital HNs, the two dominants. This one here is one. In this one, I showed you all how to do the Punnett squares, right? So these are the answers here that you're going to get, right? It should be pretty easy from seeing the answers there. So let's go on. The question isn't finished though, right? Oh, the question is finished. Sorry. They just want you to draw the diagram. So let's move on to the next question. Complete the following sentences. Meiosis is a type of cell division in which the chromosome number is how much? How much do you think? Meiosis, the one with the E, right? Did I get that correct or is that wrong? I think that one is wrong. So I have a wrong answer here, right? Meiosis has 23 chromosomes, not 46. So I made a genuine mistake there. It's a good thing I read my own answer. At the end of meiosis in humans, each daughter cell contains how much? That's all. Let me read this question properly. All right. So I think this question is stated a little bit vague. Okay, from reading this, right? Remember, I already agree with how this question is stated, but let's go through it anyway. Meiosis is a type of cell division in which the chromosome number is actually, I think they want us to put 46 here. They're talking about at the beginning of meiosis, before the divisions start. They could have expressed that a little bit better. At the end of meiosis, each daughter cells will contain half the amount of chromosomes. So it's 23. Right? So to me, I would just put a question mark by this one. I find it could have said at the beginning of meiosis to make it a little bit more clear. But anyway, let's move on. Both mitosis and meiosis occur in human cells. All right. Meiosis occurs in only one type of cell, while mitosis occurs in other types of cells. Name the type of cells in which meiosis occurs. Now, I believe I have the wrong answer here, but let's see what answer we have here, right? Oh, no, I have the correct answer. It's meiosis, sex cell. So yes, I got this one correct, right? So meiosis occurs in sex cells. That's the gametes, sperm and eggs. That's how sperm and eggs are made. If you're wondering, right, the other types of cells, remember there are many different types of cells in the body. Sex cells are very unique because they have half the amount of chromosomes, right? Sperm and eggs have half the amount of chromosomes, which is 23. All other cells are usually called somatic cells. So somatic cells are cells in the body other than the sex cells. And somatic cells reproduce or duplicate themselves by mitosis. So all the other cells in the body, with the exception of the sex cells, use mitosis to replicate. So whenever they replicate, the daughter cells still have 46 chromosomes. That's somatic cells. State two differences between mitosis and meiosis. Well, I put this in table form. What I just put here should be obvious by now. So I will let you all read those, and then you can pause the video, obviously, and let's move on. During meiosis, that's the one with the half that eventually leads to half the amount of chromosomes. Similar chromosomes pair up and exchange genes. But for those who watched part two, that's the one where I compared it to two people having paint on their hands. One had red paint and one had blue paint. And if they each touch hands, well, some paint from each of the person's hands gets swapped, right? And it's a random process of which paint will move across. You can't predict the pattern of paint being exchanged, right? Right. During fertilization, right, the normal chromosome number is attained, right? So in case, let me just review this just in case. Remember, the sperm will have 23 chromosomes, right? The egg from the female will also have 23 chromosomes. But obviously, when the two merge together, when the sperm goes inside of the egg, that's fertilization, the two chromosomes combine. So 23 plus 23 is 46. I froze there for a minute. I couldn't add there for a minute, right? However, sometimes, remember that when fertilization occurs, this is called the zygote. The zygote doesn't end up with 46 chromosomes. It will end up with 47. You all heard me talk about this condition over and over. What's the name of that condition when you have one extra chromosome? Down syndrome. So I hope you guys got that one correct. The differences among individuals are due to variation, right? Now, I explain variation is actually a good thing. Distinguish between continuous and discontinuous. We did this a few slides back. I'm not going to go into more in too much detail. My answer is on the screen there. Pause it, give it a read, and it should make complete sense by now. Let's move on. Down syndrome arises from a chromosomal mutation. How does the chromosomes of one person with Down syndrome differ from another person? Well, we just described this. Down syndrome has 47 chromosomes rather than 46. And if you want to be very, very specific, on the 21st pair of chromosomes, the 21st pair is the one that has an extra chromosome. And I believe I showed you that in part one of this series of videos, part 1 of 3. Right? You should also be seeing it in your notes. All right. So before you even start to read this, look at how they wrote the dominant and recessive genes. Usually for a dominant gene, they put a capital letter, and they're saying like capital A, and for the recessive gene, they use a common letter. But they decided to do it a little different for this particular question. They have two X's, right? So this X is one gene and this X is another gene. And then they put like a small letter here. So the one with the capital R hovering over the X is the dominant one. The one with the common R hovering over the capital X is the recessive. That's just how they wrote the question. Right, so let's read through this one. Sex-linked inheritance occurs when genes are on the sex chromosome. Where we discussed this, like red-green color blindness, right? That's a sex chromosome condition. In most cases, the genes are on the X chromosome and not on the Y chromosome. All right. Use a Punnett square to determine the genotype of an offspring of a cross between a female carrier of red-green color blindness and a normal male. Now, the normal male means that the person is homozygous dominant. So that means the male has two dominant genes, right? That is what they're trying to say, right? No, sorry. They told you what to do with this one here. Sorry, I didn't read this. The examiner is being super nice here. They said use X capital R and X common R to denote the female, while the male, right, has X capital R and then a Y with a minus sign. This means that the Y, this gene, has nothing to do with the question. In other words, it doesn't affect the sight of the person in any way, right? So X capital R is the good one, that's the dominant one, and the X common R is the one that will cause the red-green color blindness. The Y, he is like a silent person. He has no say in whether the person has color blindness or not. So think of the XY as a gene that cannot talk at all. He doesn't have a voice. So this is a kind of unique question. Let's see what we come up here with. Remember, the male, let's look at it here. The male has XR and the XY, and the female is X capital R and X common R. So these are all the different genotypes you can get, right? These are all the different combinations, right? Now, a person with XR, capital R, right? XR, both of them capital, will have normal vision because that's homozygous dominant. A person with XR, X common R, remember the X common R is the one that causes the red color blindness, but he's the recessive one. Won't the body listen to the dominant one? So this person will also have normal vision. A person with X capital R versus the XY. Remember the XY, he worse than a recessive. He don't even have a voice. He have no say in this. So the body is definitely going to listen to the dominant gene, which is normal vision. However, a person with X common R, remember he's a recessive gene. He is very weak. But the XY is even worse. He's even weaker. He has no say in this. So if a person has this color, this combination, the body is going to listen to the recessive gene in this case. And that person will have red-green blindness. Right? So I like this question. This is a very unique type of question. It's way different from the other type of questions because this one had a silent gene. And when there's a silent gene paired up with a recessive gene, the recessive gene finally wins. So let's move on. Right, some more questions here. Give it a read. This one should be very easy. Pause the video and I'm going to show you my answers. But this one, I don't think I need to read through this one, right? But these are my answers. I hope you got these answers. One of my answers is wrong. Which one is wrong there, right? Did you all figure it out? It's the third one. Mitosis occurs in somatic cells, not the sex cells. Remember, I had a note on that earlier. So the somatic cells are all the other cells other than the sex cells. So I had a wrong answer here. That was not intentional. All right, I guess I was sleepy when I was doing the solutions, right? Everybody makes mistakes. During meiosis, an event occurs in which the exchange of small parts of DNA between homologous chromosomes happen. That's the same thing with the blue paint and the red paint and two people touch hands. What's the name of that? Did you get crossing over? If you did, it is correct. Part 2, state the source of the homologous chromosomes and the importance of the events. Well, right? Remember, the source of this is the homologous chromosomes. Remember, the homologous chromosomes is a pair of chromosomes, one from your mother, one from your father. And what's the importance of crossing over, this random exchanging of genes? It allows for genetic diversity and the survival of the species. Crossing over is one way that genetic diversity is caused by, right? There are many different, there are actually, I think I gave you a list of 4 things that gives a rise to genetic diversity. Crossing over is just one of them. Right, so this one here, I have the answer for this one. I want to explain why I have the answer. In this question, part A, they ask you to define the terms gene. Now, I have to confess, in my first video of this series, I had given you all a description of what a gene is, and it's not this. If you look back to the notes on the first video, I gave you a description of what a gene does. But what a gene does is slightly different to a definition of a gene. A definition of a gene is what a gene is, not actually what it does. So in the exam, if they ask you for the definition or define a gene, I want you to remember this note in this red rectangle. You think you guys could do that for me? Because I want you to get your 1 mark. All right. So take it down. And then after that, this is going to disappear from the screen now. They ask you to define dominance. Well, we know what dominance has to do with. Dominance has to do with recessive and dominant genes. Well, when you have a pair of genes, one is stronger than the other. So dominance has to do which one is stronger and which one is weaker. The dominant one, the stronger one is called the dominant gene, and the weaker one is called the recessive gene. So here's my definition of what dominance is. That's basically saying everything I just said, but in more complex language, and you need to memorize it for the exam. Right. Part B, hemophilia, hemophilia, so I'm getting tired here. Hemophilia, right, is a sex-linked disease, right? I think we know that by now. So there's a person called Mrs. Trinbage, is a carrier of hemophilia. Explain what is meant by carrier. Now, carrier and trait is the same thing. It's like the sickle cell thing. You could carry the sickle cell disease, meaning that it's recessive, but you have a dominant gene that makes you have good blood, right? So a trait or a carrier is when a person just carries around the gene, but its body doesn't actually listen to it because there's another dominant gene that it's listening to. So that's a carrier or a trait. And I think we discussed this one already. So I have a little definition, an explanation of what a carrier gene is. By reading it, you should be able to figure it out, right? Because we did this one already. You just have to know that a carrier and a trait is the same exact thing, right? And this one here, if you notice, you have X capital H and X common H, right? Obviously, the ones with the capital H is the dominant gene. So in this one here, they're telling you the exact genotype of Miss Trinbage and the genotype of her husband. And they want you to draw up the Punnett square. That should be super easy. Pause the video and see if you can do it first. But I'm going to reveal my answer now. And this is what you would get. This is the husband here, and this is the wife for Mrs. Trinbage. And these are all the possible outcomes. I hope you got the same answer. Right. Now we're going to, now the question is continuing. So this is the same Punnett square from the last slide. And I wanted to describe the phenotypes. Remember, the phenotypes is what will happen if you have a particular set of genotypes. So let's see if you, well, pause the video and see if you can figure it out. But tell me if this makes sense. There's a 100% chance of the offspring having normal blood clotting. I can prove that. In all of these, there's at least one dominant genes. The ones that have double dominant genes, well, they are definitely going to have good blood because it's co-dominance. Even this guy here, he's a carrier of the bad gene, right? The one that makes the blood not clot properly. But there's a dominant gene that the body is going to listen to. The body is going to listen to the dominant gene, which is the X capital H, which is a good one. So he's still going to have good blood. Even this guy is still going to have good blood. But aren't these two guys carriers because they are carrying the X common H's, and maybe one day they will pass it down to their children. So they are carriers. So since there are two instances out of the four, there is a 50% chance the offspring will be a carrier of hemophilia, right? Right? There's a 50% chance, right? Because it have two out of the four. All right. All right, and that's it, guys. That's all the past paper questions. And these are the most diverse questions I could have found when I was going through the past papers. So, guys, I really, really hope part one, part two, and part three was super helpful. And I'm telling you, well, once you revise all the notes I gave you and just go through these questions, you are going to be fine for this topic, right? I can't see anything else harder coming than this, right? So, revise your notes, give it a really, really, really good revise. Just don't open it and just look at the page. Read through the notes, see if you get a blank piece of paper and write back some of the definitions. And, every couple of days, revise. Good luck, guys, for the CSEC exam. Of course, leave a comment if you feel like it. Take care.